we've heard then that enhanced vaccines can boost and improve the immune response in the elderly, and also that they give a greater breadth of protection, as Adolfo was showing us there. But what about demonstrated efficacy, and what about their ability to perhaps reduce the overall disease burden?
Thank you, and I think obviously that's what's important. If everybody in the population gets influenza and a few days later they're better and there are no complications, then we'd be comfortable with the fact that our vaccines are working. But clearly what we need is something that's going to produce these three outcomes. That we need our new vaccines to produce a higher immune response. We need the vaccines to have that higher immune response actually show an improved clinical outcome for people who develop influenza. There's a lot of debate about persistence and the breadth of protection that we get from our vaccines. I think the breadth of protection, clearly in a drifted year when our vaccines are working less well, we need to do better. And from a persistence, although there is some debate about whether a lack of persistence is due to drift in the virus during the season, change in the virus during the egg passage, and so on. I think there's enough data there to say that even in seasons where we have a reasonable match, we lose effectiveness of our vaccines as time goes on, and that's in the young as well as in the old. So what I want to do is just look at the two main enhanced vaccines that are available in the context of do they produce a better clinical outcome, do we get better breadth of protection when there's drift, and do we get better persistence over time. The first one I want to talk about is high dose, and I won't spend as much time on this because I understand at this point of time there is no high dose available in the European Union. So this is not one of the options that you have to replace your current TIV, QIV. As with other vaccines that went through a relatively standard process, they had phase 3 trials back 10 or 12 years ago which showed a higher antibody response using high dose and reduced laboratory confirmed influenza by comparison to standard TIV. It led to an accelerated approval process through VRBPAC in the US, which then came with a requirement to do an RCT to show effectiveness as part of that. And the RCT that was done showed 24% effectiveness against lab-confirmed influenza by comparison to TIV. So an increase of 25%. Probably the only worrying aspect of the RCT is that it was 35% effective against matched strains and somewhat under 20% against mismatched. So, the discussion about what happens when they drift, I'm not sure the RCT gave us confidence that it was going to work as well. But if you think back to Dan Jernigan's slide that Adolfo put up, in 2014-15 there were 950,000 hospitalisations in the United States. In that year, even a 5% improvement would have saved 86,000 hospitalisations. So in our worst case scenarios, which in fact Australia is going through this year, an improvement of 25% from a high dose vaccine is a massive benefit to the elderly, to the community and to the health system. There was enhanced protection with the RCT against serious life-threatening pneumonia associated with influenza. And when they looked at the safety profile was very similar to standard TIV. So this was a vaccine that had an improved clinical benefit in an RCT and was safe, and is now in fact about 60% of all elderly in the United States are receiving this. There was a number of very clever real-world studies that followed on from the Diaz Granados work with the RCT. And this is the first one, which was a administrative data set activity done by staff from the US CDC. They looked at about 900,000 people who'd received high-dose vaccine and 1.5 million who'd received standard vaccine. And what they found was that, again, looking at Medicaid, Medicare data from these people, 22% more effectiveness in preventing influenza than standard TIV, and 22% more effective for preventing influenza hospital admissions. So again, better at stopping influenza and better at stopping the severe consequences of influenza using the administrative data. And Stefan Gravenstein's studies in large numbers of people in nursing homes. They did a cluster randomised trial of nursing homes near CDC facilities, and what they found there, and this is obviously the toughest group to vaccinate, they're already in nursing homes, their immune system is very poor. They found that there was an overall 7% protection against hospitalisation, so all-cause hospitalisation, not flu, 7% down, and about 12.5%, 13% decreased in respiratory-related admissions. While these data were less strong than the administrative data and the RCT, this was a H1N1 year, and we expect to see decreased benefit of using the enhanced vaccines if we've got better background protection from the TIV itself. So I think at this point what we know is that from high-dose TIV, we certainly get a higher immune response and we get increased protection. And in a H3N2 year, that is very significant. At this point of time, we haven't seen data that shows that in a mismatch year that it will be as protective. I mean, the RCT, as I mentioned, didn't show the same impact against drifted strains. And so I think the jury is out at this point. As we see more of it used in the United States and Canada, we'll get a better feel for how good it is when we get drift. And again, we don't have good data on persistence because there's no publications at this point. I'm unsure whether the very early studies before the phase 3 trials had data that showed 180 or 360 day immunogenicity data, seroconversions or GMTs, but I haven't seen that. So certainly a better vaccine, but as I said, at this point, not available in Europe. For the adjuvanted TIV, the immunogenicity studies, and this has been an interesting process. I mean, the adjuvanted TIV has been available in Europe for about 20 years, and they've used 100 million doses of the product at different times, but it hasn't gone through the same initial rigour as was required to get its recent licensure in the US. And so some of these data that we're seeing actually post-date the availability of this vaccine in Europe itself, which has been there for a long time. So these are the homologous strains. So if we look at non-drifted strains and we look at the pivotal study with adjuvanted TIV, what we see is very strong data showing that there is an improvement in both the seroconversion rates and the GMTs with adjuvanted vaccine, and that improvement is most pronounced for H3N2, and happily for the heterologous strains, we see the same thing again. Not as dramatic for B, I mean only just statistically significant in the seroconversion and not for the GMTs, but very pleasing data from the point of view of protection against drifted strains, from the point of view of immunogenicity at this point of time with ATIV. Persistence data was actually looked at as part of the trials with the ATIV, and as you can see there, when we look at the one that we're most worried about, the H3N2, we have data showing an improved response at day 22, the 1 month phase. It's still there and not much reduced at day 180, and at day 365 it's still 1.3 times and statistically significant, although only just at that particular point. So again, immunogenicity data which is suggesting that with over time that we're going to get persistence of the background immunological protection using enhanced vaccines. And these data are supported by the Ansaldi study that was put up looking at what happens with drifted strains in the mid-2000s. and it's supported by a lot of the work that's gone on with pandemic planning. We had to put an adjuvant to the H5N1 to get the amount of antigen down so that we had enough vaccines. Otherwise, we were using 75 micrograms. Recent studies with H7N9 found candidate vaccines that only really worked in 2 studies when the MF59 was there. So, there's good data that suggests that the immunogenicity, the theoretical processes that Adolfo was describing to us actually produced an enhanced immune response in people. This is the safety profile, and obviously one of the things that people get worried about when we say, hey, this vaccine's much better is, is it going to produce more side effects? Am I going to worry about how safe it is? And you can see from this that the comparison of the serious adverse effects and death and the others was pretty much identical. The data from solicited adverse events was higher for ATIV, but the vast majority of those were in fact self-limiting local reactions, and we use this a lot in my time in Canada. People got more of a sore arm than if we gave them standard vaccine. I used to tell them without any scientific basis at all that was a good thing. It showed that the vaccine was working and that they should be pleased about having a sore arm. No, and my green button has got obsolescence instead of immune senescence.
Perhaps
Can
we
I
could
try the
call
other
on
one?
a little technical help here. So as you say, you're very comfortable then about the safety profile
I'm
here.
very comfortable. There was a number of other studies which weren't part of the pivotal trial, and in particular David Shifley's group in British Columbia did a study of immunogenicity, acceptability, tolerability of 3 vaccines, standard TIV, the intradermal vaccine which was available in Canada at that time, and the adjuvanted vaccine. And what they found was that there was an increased pain reaction from adjuvanted vaccine, but that 96% of people would go back and have it the next year. So this wasn't something that wanted people to not have that vaccine. It was something they accepted as being reasonable for that.
Sorry, George, do you want to come in there?
Yes, Paul, I wonder whether if in these studies the vaccine was to be given with the airlock technique, whether we'll get less local side effects.
I think, I haven't seen data, but I'm not surprised about the local side effects, because if we think back to what Adolfo was saying about the mechanism of action, the adjuvant works to increase the number of APCs that are arriving at the site. There's a greater dendritic response, and so that there is a local reaction prior to transfer to the lymph nodes that I expect to be bigger than if I'm using TIV. Yeah. And so the mechanism of getting a better response is associated with more reactogenicity at the local site, but not with serious side effects. Okay, I want to talk quickly about 2 real-world studies, because I think this is what's important to me. I have 53 nursing homes in the area that I look after, and they're the sort of people who don't make it to an RCT. RCTs are interesting, but I want to know if this vaccine works in the real world. This was the Lombardi study, which ran over 3 years. It wasn't randomised in that the physicians made decisions as to who they wanted to give the vaccine to. Large numbers of people, over 100,000 people enrolled over that period of time, some of whom had adjuvanted vaccines, some of whom had normal TIV. And because it wasn't standardised, what we saw was that there were actually 2 different populations. And when we looked at the people who had adjuvanted vaccine prior to winter, they were 17% more likely to be hospitalised. So these are people with more comorbidities. They were sicker. And decisions were being made by the Italian primary care physicians that they thought the adjuvanted vaccine might be better, so they gave it to the people who were sicker. And so we started with a group that said, I'm giving the adjuvanted vaccine to people who are more likely to be hospitalised anyhow. And what we found afterwards was that there was a 25% reduction in hospitalisation due to respiratory disease with people who had adjuvanted vaccines. So that 25% is almost certainly an underestimate of the improved attributable enhanced protection from adjuvanted vaccines, because they were a group that we were expecting the vaccine wouldn't work as well. So that was very positive. And then this was the next of the community-based test negative control studies. It was the British Columbia study. We gave adjuvanted vaccine to the elderly in 2 health authorities, and in the other health authority they got TIV. It was a challenging year. It was a very low influenza season. There were swabs taken. So this had the benefit of a single lab, very controlled laboratory process, a public health unit that had access to everybody's results, and a process where we could check every single person who had the vaccine to look back. And rather than hospitalisations for pneumonia or anything else, this was lab-confirmed PCR-based influenza. So, much different from the point of view of the sensitivities that we were talking about. And these were the data, and these data are in fact more dramatic than many of the others. And it's largely because this was a year that TIV was relatively ineffective. So what we found was that on a corrected basis, the overall adjuvanted vaccine was about 58% effective, and the TIV was not effective at all in that particular year. And what we got between the two of them was that the relative attributable vaccine efficacy was way up at 63% for adjuvanted over TIV. And as I said, these are the issues when you start to get TIV not working, you enhance the benefit of using adjuvanted vaccines in that particular group. This is the last slide that I want to have a look at. In our BC study, we didn't compare it to elsewhere because we had our BC study. And what we found in the second year when there was an improvement in TIV was that our adjuvanted vaccine was about 63% effective in preventing hospitalisation, which was much higher than the national data on TIV. This is a study done by Shelley McNeil's Serious Outcome Surveillance Group, looking at the course of 3 years, looking at non-adjuvanted versus adjuvanted vaccine. Again, you can see that there are large confidence intervals in these data, but these data mimic the data from the British Columbia study in the 2 years earlier than that. And what they say is that there's about a doubling in the protection using an adjuvanted vaccine against hospitalisation. And this is important because when you look at the BC data, over half the people were over 85, half were in long-term care facilities. And these are the groups that don't respond to vaccine, and they're the group that are having increased protection with adjuvanted vaccines. So, I think the message from this is that the adjuvanted vaccine clearly has an improved immune response, clearly has an enhanced benefit from clinical protection. There are data, but they're immunogenicity data, that suggests that the breadth of protection is better through a range of studies from H5N1, H7N9 to the Ansaldi work in the drifted 2013-14 year between A Texas and A Hong Kong and so on. Clearly, at some point, we'd like to see some enhanced breadth of protection with effectiveness, not just immunogenicity. And similarly for persistence, we have good immunogenicity data saying this stuff lasts the whole year. We now need to sort of roll that into saying, let's get some effectiveness data. This vaccine is available in Europe. It's available in many countries. It is greatly underutilised given the potential benefit for the elderly that we're seeing with this.
Thank you very much, Professor. Well, let's pick up that point about the underutilisation of the enhanced vaccines. Perhaps, George, I can put that to you. Is that valid in other countries? Is it valid in the UK where you have experience?
Well, we don't yet have the enhanced vaccines. We only have the usual TIV and QIV, and also for the children we have the intranasal vaccine, which is a completely different vaccine because it is a live vaccine. Mm-hmm. What we do need is to actually improve the effectiveness of the vaccines we give, particularly for our patients 65 years and over. So if we can actually have more effective vaccines, then we'll be delighted. And the JCVI has actually given an indication that they want to review the vaccine effectiveness for the elderly now and not into year 2020, because they say there are vaccines in the horizon that can do so much better than the ones we have at the moment.
And the point also that Paul made in there about the in drift years and whether there's data yet to show the impact of these. Adolfo, is that an area that you can comment on, that where there's drift, the mechanism of action that you were describing, do you think that that has an impact?
Well, the vaccines that we use right now, the inactivated vaccines, they work mainly by inducing neutralizing antibodies against the hemagglutinin. And the neutralizing epitopes that they recognize, they are the ones that change every year due to pressure, selective pressure of these antibodies against the virus, and that generates drift. So when the virus drifts, it's evading a neutralizing antibody response that has been induced either by natural infection in the past, and now also on the top of that, vaccination. But the virus cannot change dramatically. We're not talking about the pandemic, but there is an antigenic shift. The drift is progressive. Mm-hmm. And it could be some years more drift or less drift, depending on how the changes are affecting the antigenicity. But certainly there is, even in the drifted strains, there are still some neutralizing epitopes that are shared with the non-drifted strains when there is only 1 year of difference or 2 years of difference. So a more repertoire of antibodies against the neutralizing epitopes of the hemagglutinin results in a better neutralizing response against drifted strains. And the more stimulation you get of multiple B-cell lineages, the better you're going to come out with a more breadth of protection, more breadth of neutralizing antibodies that recognize multiple neutralizing sites in the hemagglutinin, that are some of which still remain the same even during drifted strains. The problem is that if you get only antibodies against a very specific epitope, then when the drift happens, then you lose the protection. And that's, I think, what some of the new vaccines that they are now being used, adjuvant seems to induce a much breadth of these antibodies against multiple epitopes, some of which are shared with the drifted strains.
And so as we're hearing then that the enhanced vaccines are not available everywhere globally yet, but where they are, Paul, what are the factors that a clinician should consider when they're deciding which immunisation to recommend, where they do have a choice?
I think we need to do a better job at educating primary care clinicians about the severity of influenza. I still have people who recommend pregnant women don't get vaccinated, which makes me pull my hair out. I have people who say it's not important for children, and I have people who say influenza is not a serious disease and vaccine doesn't work. I have an occasional doctor that still thinks a couple of bits of protein can give them the flu. And so, there's a real issue here that one of the things we need to do is make sure that our primary care clinicians are saying influenza is a nasty disease. It's a particularly nasty disease in the elderly, and we must use whatever vaccines that we have. If you only have TIV or QIV, in the elderly they're actually much of a muchness. The potential benefit of adding a second lineage of influenza B to the elderly is much, much less than in children. And so if you only have QIV, like in Australia, use QIV. I think it's obviously better than TIV because it's a little bit more breadth, but it's a relatively ineffective vaccine with more breadth. If that's all you have, you use it because you'll still get benefit, and in a non-drifted year you'll get good benefit. It may only be 40%, but what I'm seeing this year in particular, and we saw in the US last year, was that if you get influenza after having had the vaccine, you're more likely to have a milder case. One of the comments from CDC, from Bill Schaffner this year was, you still got the flu, but you weren't in hospital and you didn't die.